students, today we will learn fat. Fat belongs to a chemical group called lipid. Fat is an essential part of our diet. It contributes 10 to 30% of total energy need, and fat is also important part of our body structure. All cells and tissues have fat as integral part. Vital organs such as heart, liver, brain are protected by a sheet of fat and water which keep them in place and protect from physical injury. Nerves are also protected by fat. A layer of fat beneath the skin acts as insulating material against cold. Fats around our joints acts as a lubricating agent and allow us to move them smoothly. Therefore, fat is an important part of our body composition. Food fats include solid fat, liquid oil, and related compounds such as fat-soluble vitamins and cholesterol. Lipid is overall group which contains all fats and related compounds. Lipid has been derived from a Greek word lipos. Lipos means fat. It is a complex organic compound which are insoluble in water but soluble in certain solvents such as petroleum ether, diethyl ether, benzene, hexane, etc. They occur widely in plant and animal kingdom in the form of oil and fats respectively. They are important part of our body structure. It accounts 15 to 20% of total body weight in healthy non-obese men and 18 to 25% of total body weight in healthy non-obese women. It is a complex organic molecule constituting a mixture of fatty acid and alcohol, generally glycerol. Like carbohydrate, it is made up of carbon, hydrogen and oxygen. But it differs from carbohydrate in that it contains more carbon and hydrogen and less oxygen molecules. Therefore, more oxygen is required to oxidize fat. Therefore, more energy is liberated when fat is oxidized. 1 gram fat gives 9 calories, which is 2.25 times higher than that of carbohydrated protein. Based on the chemical structure, lipids have been classified as simple lipids, compound lipids, derived lipid and miscellaneous lipids. Simple lipids are those lipids which contain fatty acids and alcohol. Compound lipids are those lipids which in addition to alcohol and fatty acid, it contains other substances. Derived lipids are derived from simple and compound lipids. Miscellaneous are those which possess characteristics of lipid. Based on the nature of the alcohol, simple lipids are classified as fats and oil and waxes. Fats and oils are triglyceride. They are made up of 1 molecule of glycerol and 3 molecules of fatty acid. And fats are solid at room temperature while oils are liquid at room temperature. Waxes are ester of fatty acid with long chain aliphatic alcohol or cyclic alcohol, mostly cetyl alcohol, and they include true waxes, cholesterol esters, vitamin A esters, vitamin D esters. As I said earlier, compound lipids are those lipids which in addition to alcohol and fatty acids contain other substances. They are different types. Phospholipid, it contains phosphoric acid, nitrogenous base in addition to fatty acid and glycerol. Glycolipids are complex lipids containing carbohydrate in combination with fatty acid and sphingosine. Sphingosine is 18 carbon atom amino alcohol with unsaturated hydrocarbon chain. Sulfolipid, which contains sulfuric acid in combination with hexose in a cerebroside. Lipoprotein, lipid is bound to protein. Derived lipids are those which are liberated during hydrolysis of simple and compound lipids, and they retain the properties of lipids such as fatty acid, glycerol, diglyceride, monoglyceride. Miscellaneous are those lipids which possess characteristics of lipid, example steroid, terpenoids, carotenoids. Simple lipids, they are made up of fatty acid and glycerol. When they are hydrolyzed, they yield 1 molecule of glycerol and 3 molecules of fatty acids. Fatty acid occurring in natural fats and oil contain even number of carbon atoms. And simple lipids are neutral lipids, and they account 98 to 99% of food and body lipid. This is fatty acid. Fatty acid is the building block of fat. Fatty acid of biological importance are straight chain monocarboxylic acids with even number of carbon atom. The structure of fatty acid is given. It has carboxylic group at one end and methyl group at the other, and the chain of carbon and hydrogen in middle. About 40 fatty acids are found in nature. General formula of fatty acids are CH3, CH2 and COOH, where N can be any even number from 2 to 24. The fatty acids have 3 important aspects: chain length, saturation and essential fatty acid. The number of carbon atom in fatty acid decide the chain length, and depending on the chain length, fatty acid can be classified as short chain fatty acid, medium chain fatty acid and long chain fatty acid. Short chain fatty acids contain 2 to 8 carbon atoms. Examples are butyric acid, caproic acid, caprylic acid. Medium chain fatty acids contain 10 to 14 carbon atoms. Examples are capric acid, lauric acid, myristic acid. Long chain fatty acid contains 16 to 24 carbon atoms. Examples are palmitic acid, stearic acid, oleic acid. Based on the nature of the hydrocarbon, fatty acid can be classified as saturated and unsaturated fatty acid. Saturated fatty acids are those fatty acids which do not contain any double bond. They contain single bond, and therefore carbon is saturated with hydrocarbon. Example is stearic acid, palmitic acid. Unsaturated fatty acids are those fatty acids which may contain one or more double bond. Unsaturated fatty acids are further subdivided into monounsaturated fatty acid and polyunsaturated fatty acid. Monounsaturated fatty acids are those fatty acids which contain one double bond. Example is oleic acid, it contains one double bond. Polyunsaturated fatty acids are those fatty acids which contain 2 or more double bond. Examples are linoleic acid, it contains 2 double bond, linolenic acid which contains 3 double bond. Now we will study nomenclature. Numbering of carbon atoms in a fatty acid can be done from both sides. If it is done from carboxylic end, then the carbon atom of the carboxylic group is given first number, second will be 2, 3 and so on. Carbon adjacent to carboxylic group is labeled as alpha, this will be beta, gamma and so on. If numbering is done from methyl end, it is also known as omega end, then the carbon atom of the methyl group will be given omega 1, this will be omega 2, omega 3 and so on. Similarly, the position of double bond can also be indicated in 2 ways. If take an example of double bond between carbon atom 12 and carbon atom 13. If counting is done from carboxylic end, then it can be reported as C12 या delta 12, and if counting is done from methyl end, it will be reported as omega 6. In both the cases, beginning of the double bond is taken, and in saturated fatty acid, suffix anoic is added to systematic name of fatty acid, and in unsaturated fatty acid, suffix enoic is added to the systematic name of the fatty acid. Now come to the omega classification of fatty acid. As I discussed, the methyl end is also known as omega end, and if the first double bond is in the third carbon atom from the methyl end, it is called omega 3 fatty acid. If the first double bond is in the sixth carbon atom from the omega end, it is called omega 6 fatty acid. If the first double bond is in the ninth carbon atom from the methyl end, then it is called omega-9 fatty acid. An example of omega-3 fatty acid is alpha-linolenic acid, EPA, DHA. We will discuss them in subsequent slide. Example of omega-6 fatty acids are linoleic acid, arachidonic acid, and example of omega-9 fatty acid is oleic. This table shows the important natural fatty acid and their occurrence in food. Acetic acid, butyric acid, caproic acid, caprylic acids are short chain fatty acids. Myristic acid is medium chain fatty acid. Palmitic and stearic acid are long chain saturated fatty acid, and they are widely found in food lipids. Oleic is monounsaturated fatty acids. Linoleic acid is polyunsaturated fatty acid. Alpha-linolenic acid, arachidonic acid, EPA and DHA are also long chain polyunsaturated fatty acid. In the column 2, this is the shorthand representation of the fatty acid. The shorthand notation of the fatty acids have 2 letters separated by a column. Take an example of stearic acid. Stearic acid is made up of 18 carbon atom and it is saturated. The first letter indicates the number of carbon atom and second letter indicates the presence of double bond. Since it is saturated fatty acid, it does not contain double bond, therefore 0 is written here. Take an example of unsaturated fatty acid, linoleic acid. It is made up of 18 carbon atom. It has 2 double bond, and from the omega side, the first double bond is in the 6th carbon atom, therefore N6 is indicated here. What are the essential fatty acids? Essential fatty acids are those fatty acids which can't be synthesized in our body. Therefore, they must be supplied through the diet. Essential fatty acids are 2 types, linoleic acid and alpha-linolenic acid. Apart from this, arachidonic acids are considered semi-essential fatty acid because it can be synthesized in our body from linoleic acids. They play important function in our bodies. They are essential for growth in youngs. They are required for maintenance of the normal healthy skin. They are components of membrane. As components of membrane ensure their permeability to water and small molecules. They are precursors of eicosanoids. Eicosanoids are a group of important metabolites which regulate vascular function in our body. One of these are prostaglandin. It protects us from cardiovascular disease. Essential fatty acids have been shown to decrease the plasma level of triglyceride, bad cholesterol. It reduces high blood pressure, and also it reduces the atherosclerotic deposition of fatty material on the wall of the arteries. Now we will learn sources of essential fatty acid. Safflower, sunflower, Niger seed, cotton seed, linseed, corn, walnut, sesame, soybean oil are rich source of essential fatty acid, and these oils must contain 50% of the fat in the diet. Rapeseed, groundnut and rice bran oils are good source of essential fatty acids. Egg yolk fat, poultry fat and pig body fat are fair sources of essential fatty acid. While coconut oil, palm kernel oil, vanaspati, animal fat, butter and ghee are poor source of essential fatty acid. Now, I already discussed what is omega-3 acid. Omega-3 fatty acids are those fatty acids which contain first double bond in 3rd carbon atom from the omega end. Dietary omega-3 fatties are 3 types: alpha-linoleic acid, eicosapentaenoic acid, docosahexaenoic acid. ALA can't be synthesized in our body, but our body can form EPA and DHA from ALA. The rate of conversion of ALA to EPA is 5 to 10%, whereas in the rate of conversion from ALA to DHA is 2 to 10%. EPA and DHA are more biologically active as compared to alpha-linolenic acids. Now we will learn the functions of omega-3 fatty acid. Omega-3 fatty acid reduce low density lipoprotein cholesterol, therefore they are good for heart. They stimulate blood circulation. Persons with problem of blood circulation such as in varicose vein, as it stimulates blood circulation, it is beneficial and it helps to break down clot. It also helps to reduce blood pressure and blood triglyceride. Therefore, essential fatty acids help in reducing both primary and secondary risk of cardiovascular disease. It is good for the person suffering from rheumatoid arthritis because it reduces tenderness in joints and also reduces the need of corticosteroids, and they have important role in fetal brain and eye development. Therefore, adequate maternal intake of essential omega-3 fatty acid must be ensured in the time of pregnancy to meet the increasing need of the growing fetus. What are the foods which contain omega-3 fatty acid? Alpha-linolenic acid can be obtained from green leafy vegetable, nuts, vegetable oils like canola oil, soybean oil, flax seed, mustard seed and rapeseed. EPA and DHA is found in certain oily fish like salmon, herring, mackerel, sardine and tuna. They are also found in walnut and oil of brown. This table shows fatty composition of fats and oil. Coconut, ghee, banaspati, palm kernel are rich in saturated fatty acid. They contain saturated fatty acid to an extent of 60 to 90%. Monosaturated fatty acids are found in red palm oil, groundnut, rice bran, sesame and olive oil. High amount of linoleic acids are found in sunflower, safflower, sesame, corn and cottonseed. And alpha-linolenic acid can be obtained from the mustard, rapeseed, soybean, canola to an extent of 5 to 10. We will learn types of lipid present in food as well as in human body. Triglyceride, they comprises about 95% of food lipid and body lipids. Triglycerides are a storage form of fat in our body. When we eat a high calorie diet, then surplus energy is converted into triglyceride and stored in our body. Phospholipids is present in every cell and they are important as a component of active tissue like brain, nervous tissue and liver. Their structure is similar to triglyceride, but they contain phosphate attached to glycerol and fatty acid in place of one fatty acid. Most common phospholipids is lecithin, which is an important component of the cell membranes. Other phospholipids are sphingomyelins and cerebrosides. They are found in brain. Phospholipids are powerful emulsifying agent and they are essential for digestion and absorption of the fat. Cholesterol is a word we come across very often in the advertisement of the fats and oils in respect to heart health and high blood pressures. Cholesterol is essential lipid. It is synthesized in the liver independent of dietary intake and it can be synthesized about 2 grams per day, which is circulated in the bloodstream and used wherever it is needed. It is precursor of bile acids, vitamin D and hormones. Hormones which are made up of cholesterol are sex hormone and adrenal hormone. Cholesterol is present only in animal kingdom. They are not present in plant food. The foods which are rich in cholesterol are mutton, pork, ham, sausage, lamb, chicken, eggs, whole milk, cheese, ice cream, butter and desi ghee. And the normal blood level of cholesterol is less than 200 mg per dl. If it is more than 200 mg per dl, then the condition is known as hypercholesterolemia, which is the increased risk of coronary heart disease. Lipoprotein, lipid is bound to protein. It is synthesized in the liver. Main function of lipoprotein in our body is to transport fat. And depending on the density, it is grouped into chylomicron, intermediate density lipoprotein, very low density lipoprotein, low density lipoprotein and high density lipoprotein. Low density lipoprotein bound cholesterol is the primarily responsible for atherosclerotic buildup of lipid material on the walls of arteries. and HDL is responsible for reducing this buildup. Therefore, LDL is called bad cholesterol and HDL is called good cholesterol. And like cholesterol, lipoprotein level in the blood is routinely checked to detect the lipid disorders which is related to the coronary heart disease. Now we will learn properties of fat. As you might be knowing that it is insoluble in water, but they are soluble in some organic solvents like ethyl ether, petroleum ether, benzene, hexane, chloroform. Saponification value or saponification number, it is measure of average molecular weight fatty acid in the food. If value is high, then molecular weight will be low. It is defined as number of milligram of KOH required to saponify 1 gram of fat or oil. Iodine value is the measure of extent of unsaturation in fat, and it is defined as number of gram of iodine absorbed by 100 gram of fat. Rechet-Mitchell value is defined as the number of milliliter of 0.1 alkali NaOH or KOH required to neutralize the steam volatile water soluble fatty acid present in 5 gram of fat. This test determines the amount of butyric acid and caproic acid which is readily soluble in water, and a part of caprolic acid which is slightly soluble in water. Acid value is the measure of free fatty acid, and it is defined as the number of milligram of KOH required to neutralize the free fatty acid in 100 gram of fat or oil. The value is expressed as percent of oil. Density in fat. Density means development of off flavor, and development of off flavor is often accompanied by change in the texture and change in the appearance. There are three main types of rancidity which is found in fat are hydrolytic rancidity, oxidative rancidity, and ketonic rancidity. Hydrolytic rancidity is due to the lipase enzyme, and this rancidity need not always produce off flavor. Off flavor is produced only when the small molecular weight fatty acid like butyric acids and others are released by the action of the lipase enzyme. And higher fatty acids such as palmitic and stearic acid have very little odor. They do not contribute to the off flavor of the fat. Oxidative rancidity is very common. The oxidation takes place at the unsaturated linkage. The addition of oxygen to unsaturated linkage results in the formation of peroxide, which on decomposition yields aldehyde and ketones, which contributes to off flavor. Certain metals like copper, iron, zinc, they accelerate the onset of oxidative rancidity. Ketonic rancidity is due to the contamination of fat such as coconut oil with certain fungi, Aspergillus niger, Penicillium glaucum. The off flavor developed may be due to the production of aldehyde and ketones by the action of the enzyme present in fungi on the oil. Moisture accelerates ketonic rancidity. Now we will learn functions of fat. You must be knowing few of them because I have already discussed them. Fat is concentrated source of energy. 1 gram provides 9 calories, which is 2.25 times higher than that of carbohydrate and protein. Fat, they are source of essential fatty acid and fat soluble vitamins. Fat is essential for the absorption of fat soluble vitamins A, D, E, K and carotenoids. Cholesterol is an essential lipid, and certain hormones and bile acids are formed from cholesterol. Phospholipids are important component of plasma membrane. Flavor, palatability, satiety value can be enhanced by fat, and due to slow rate of digestion of fat in the stomach gives us a feeling of fullness. Fat deposited in adipose tissue serve as a reserve source of energy during starvation and illness. Vital organs such as brain, heart, liver are protected by a sheet of fat and water which hold them in place and protect from injury. Since fat is a bad conductor of heat, The layer of fat beneath the skin acts as an insulating material. They conserve heat and regulate body temperature. Important sources of fats are pure oil and fats, ghee and vanspati. They contain 100% fat. Butter contain 80 to 81% fat. Among the good sources, nuts and wild seed, milk powder, eggs, meat and fish, and they contribute 14 to 60% fat in our diet. Cow's milk, buffalo's milk, pulse, whole cereals and millets are the fair source of fat in our diet. RDA for fat is given in this table. Depending on the physical activity, 25 to 40 gram visible fat per day should be consumed by man, and 20 to 30 gram per day has been recommended for the adult women. During pregnancy and lactation, 30 gram per day has been recommended, and for children 27 to 30 gram has been recommended. For adolescent boy, 35 to 50 gram per day has been recommended, and for adolescent girls 35 to 40 gram per day has been recommended. It is the recommendation of vegetable fat. Vegetable fats are those fats which we use in cooking or we eat as such as butter and ghee. Hydrogenation. Hydrogenation is a process by which liquid fat is converted into semi-solid and solid fat for use as a shortening in the production of bakery items. Hydrogenated fats have long shelf life, and since the deodorized refined oil is used to produce the fat, the resultant fat is also colorless and odorless. Therefore, they can be used in several preparations. Hydrogenation is carried out at high temperature, 300 to 370 degrees centigrade under pressure in the presence of nickel catalyst for 1 to 3 hours till the desired consistency is obtained. Hydrogenation causes production of trans fatty acid. We will read about trans fatty acid in this slide. Trans fatty acids are unsaturated fatty acids having at least one double bond in trans configuration. Fats in the food is in cis form. They have hydrogen atom with respect to double bond on the same side of the molecule. So they have V set. But trans fat, they have hydrogen atom on the opposite sides, and they assume a linear configuration like saturated fatty acids. This change in configuration alters the physical properties of the oil. Oleic acid, which is cis isomer, is liquid at room temperature, whereas its trans isomer, ellaric acid, is solid at room temperature. Hydrogenated fat contains ellaric acid. There are certain trans fatty acids are also produced naturally in the stomach of the ruminant animals by the action of the bacteria. Therefore, the ruminants products like meat and dairy products, they also contain a small amount of trans fatty acid, and major isomer found in ruminant fats are vaccinic acids. This hydrogenated major contributor of trans fatty acids in our diet is partially hydrogenated vegetable oil, and they are used widely because they have long shelf life, they are stable during frying, and their solidity can be customized to enhance the flavor and palatability of the food. Major contributor of trans fatty acid in our diet is bakery items, packaged snack, and deep fat fried fast food. Essential fatty acid deficiency in human beings causes dry and scaly skin and itching sores on the scalp and growth retardation. A skin condition known as phrenoderma, in which skin becomes thick, rough, and horny papules of a size of pinhead erupts in certain parts of our body, notably on the posterior and lateral aspects of the limbs, on the back and buttocks, and these are cured rapidly by the administration of linseed or safflower seed oil, which is rich in essential fatty acid, along with vitamin B complex. Fat is essential, but too much fat is harmful. They lead to a number of heart disease. Energy intake in excess of our need, it leads to obesity and unacceptable high lipid profile. If unchecked, there is deposition of fatty material with formation of plaque on the wall of arteries, which disturbs the movement of nutrients and oxygen to the cell. And it leads to a variety of heart disorders such as atherosclerosis, angina pectoris, blood pressure and others. Increased blood cholesterol above 200 mg per dl, LDL above 130 mg per dl, triglyceride above 200 mg per dl are associated with increased risk of coronary heart disease. LDL is beneficial because it is involved in reverse transport of fat. It carries free cholesterol from the cell to the liver for its breakdown and excretion from the body. Therefore, LDL is called good cholesterol. Diet-related chronic disease can be prevented by establishing healthy lifestyles. I have suggested some points. Control total energy to maintain ideal body weight with reference to your body build and height. Fat should be eaten in moderation. Reduce the intake of saturated fat because saturated fats are known to enhance the production of cholesterol and LDL in the liver, which lead to increased level of these lipids in the plasma. And saturated fats are mainly obtained from the animal source, so we should limit the intake of saturated fat, and the saturated fats are found in ghee, butter and meat products. Reduce food cholesterol intake to 200 mg or less, which can be achieved by restricting the cholesterol rich foods such as ghee, fat, meats, and also consuming foods like skimmed milk, toned milk, double toned milk in place of full cream milk. Diet should provide more PUFA and MUFA. Moderate the intake of omega-6 polyunsaturated fatty acid and increase the intake of omega-3 fatty acid. Limit intake of commercial bakery products, deep fried products and ready to eat products because they are major contributor of trans fatty acid, which is most atherogenic among all the fats. Use of 2 or more vegetable oil is recommended because dependence on single oil will not ensure the optimal intake of various fatty acids. Therefore, blend of oils should be used for daily cooking. And with this, fat chapter is over. Thank you.